The use of an advanced imaging technique has revealed previously unseen intricacies within peptide structures. Greetings, everyone! Today we will be delving into an intriguing subject in our video. It revolves around the groundbreaking development of a cutting-edge imaging method known as single molecule orientation localization microscopy, SMOLM. This technique has been specifically designed to explore cross-B fibril assemblies, which hold great significance in the realm of designer biomaterials. By utilizing Nile red, researchers are able to visualize the intricate structures that form as a result of synthetic peptides and amyloid beta. Through this study, fascinating disparities between the stacking patterns of synthetic peptides and amyloid beta have been unearthed, shedding light on the process of protein stacking and its potential therapeutic applications. This innovative approach has opened up exciting new possibilities for the examination of biological structures on the nanoscale. So, without any further delay, let us embark on our captivating discussion. The utilization of cross-B fibril assemblies as fundamental components in the development of custom biomaterials for medical purposes is highly advantageous. However, there is a disconcerting similarity between these assemblies and the amyloid beta structures associated with neurodegenerative diseases. Scientists aim to understand the relationship between different peptide sequences and their diverse levels of toxicity and functionality. This exploration encompasses both naturally occurring peptides and their artificially engineered counterparts. Thanks to a successful partnership between Matthew Liu, an associate professor in the Preston M. Green Department of Electrical and Systems Engineering, and Jai Rudra, an associate professor of biomedical engineering at WashU's McKelvey School of Engineering, scientists now have the ability to closely examine fibril assemblies. This examination has revealed significant disparities in the stacking patterns of synthetic peptides when compared to amyloid beta. Liu emphasized the importance of engineering microscopes to improve nanoscale measurements, thereby facilitating the advancement of scientific research. Liu and his team detailed their utilization of the Nile red chemical probe to illuminate cross-B fibrils in a recent publication in ACS Nano. Their method, known as single molecule orientation localization microscopy, SMOLM, employs the illuminating bursts of light emitted by Nile red to visualize the intricate fiber formations created by both synthetic peptides and amyloid beta. Here's the recreated text. It turns out that these assemblies are far more intricate and diverse than initially expected. However, this is actually a positive development, as it indicates that there are multiple methods for safely arranging proteins. By obtaining more precise measurements and visual representations of fibril assemblies, bioengineers can gain a deeper understanding of the principles that govern the impact of protein structure on toxicity and biological activity. This knowledge will ultimately contribute to the development of more efficient and less harmful therapeutic treatments. The challenge for scientists lies in discerning the distinction between them, a task made difficult by the minuscule size of these assemblies. According to Liu, the extremely small size of these fibers makes it impossible to detect their helical twist using an optical microscope or even certain super-resolution microscopes. Thanks to the advanced imaging technology that Liu's lab has been working on for the past few years, they now have the capability to observe and distinguish the variations. In the realm of fluorescence microscopy, fluorescent molecules serve as the illuminating source to draw attention to specific elements within a biological subject. In this particular study, the researchers employed Nile red, one of these fluorescent probes, as a means of detecting its surroundings. As Nile red navigates its environment in a haphazard manner and encounters the fibrils, it emits bursts of light, which can be quantified to ascertain the location and alignment of the fluorescent probe. Utilizing this information, the researchers can construct a comprehensive understanding of engineered fibrils that exhibit distinct stacking patterns compared to naturally occurring ones, such as amyloid beta. On the cover of ACS Nano, a captivating image of fibril assemblies was presented. The talented Weiyan Zhou, the first author, skillfully arranged the image by assigning colors to the Nile reds, resulting in a mesmerizing depiction. The final image showcases a flowing assembly of peptides, resembling a picturesque river valley with hues of blue and red. Their intention is to persist in the advancement of methodologies such as SMOLM, with the aim of expanding possibilities for exploring biological structures and processes on the nanoscale. According to Liu, we are witnessing phenomena that are currently beyond the capabilities of existing technology to detect. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.